President Lal Mosa is unable to join just yet because he is in the middle of a power cut. So let me introduce our two speakers, Hasula Wickremasinghe and Tharaka Muthunayake, who are going to do a joint presentation. I'll introduce Tharaka first. He is a teacher by profession, freelancing at the moment, graduate of the University of Colombo with an honors degree in zoology, specialized in wildlife management and conservation. And his undergraduate research project was on the phylogenetic analysis of P.bandula. Specialized in wildlife management and conservation. And then he went on to do, his research project was phylogenetic analysis of the P.bandula, which is the subject of today's talk. And that took him to join the conservation program for this fish in 2010. And he has been working on that project up to date. So he's also had a training stint in India, where he used his experience in designing the community awareness program at Galapitamada, which is where this fish was a new site. This fish was introduced, so he will talk about that. He is also a member of the fish expertise group in the Red List 2012, and he has worked as a biology teacher in St. Joseph's College, Colombo 10, and now he is freelancing. Now, Hasula, who I have known for a great many years because we have worked together on various projects when she was in the Biodiversity Secretariat. She has a BSc from the Open University and an MSc from the University of Colombo, has studied the ecology of the Bandula barb for her MSc project, and has continued the conservation work by developing the recovery plan and conducting the translocation and monitoring and the awareness work up to the present. She has also obtained a postgraduate diploma in archaeology from the University of Ketala. She has an interest in paleobiodiversity of Sri Lanka, fossil records. And she has worked for many years at the Biodiversity Secretariat of the Ministry of Environment, for 10 years. And she's currently working at the Climate Change Secretariat as a program assistant. And I am waiting for her to get back to the Biodiversity Secretariat, because when she was there, she was instrumental in getting me to do some publications, one on marine shells and another, and a poster. And we were working on some other projects by the time she left. So without more ado, I will now invite Hasula and Tharaka to make their presentation about the conservation of the fish P.bandula. Hasula?
Yes, thank you, sir. So to begin, I would like to thank the Sri Lanka Natural History Society, especially Dr. Malik Fernando, for inviting us to present our findings on the Bandula barb and respective conservation activities. So this will be a joint presentation by myself and my colleague Tharaka Muthunayake. So the slide shows the core research team which worked on the Bandula barb and is still working on it. And Professor Devaka Veerakon was my supervisor for the MSc project from 2003 onwards, and continued to guide and supervise the recovery plan, translocation program, as well as the community development program. Next slide, please. To introduce the Bandula barb was first noted as a new species by Mr. Ranjit Bandula. He then notified Mr. Pethia, and Mr. Hapugoda and Ketala described the P.bandula as a new species in 1991. It's commonly known as the Bandula Barb or else the Giantia. It's called the Giantia because when the sun shines on the barb, it shows a lovely hue of green, pink, and various other colors, which are quite close to the Buddha Giantia flag. It's an endemic fish, and it was listed as a critically endangered fish in the red list of 2012 and 2020. Next slide, thank you. The P.bandula, to identify, you can easily see because it has two vertical stripes, and then the
Hasula, we can't hear you now. Hasula, we can't hear you.
Yeah, might have been some connectivity issue, no? I think she got disconnected.
right, sorry.
All right, you're back, yes.
And the female, as you can see from the below picture, it can be identified because the dorsal fin is half hyaline. And the fish has an incomplete lateral line which perforates to 8 to 11 scales. And the amazing thing is there's also an extra row of scales above the lateral line. And these can all be observed, with your naked eye when the fish gets close. Next slide, please. Of course, you can easily get mixed up as well, because probably that is why it took some time to identify the fish as a new species, because it also gets mixed up with Pethia nigrofasciatus or the bulat hapea. But of course, it has 3 vertical stripes. And it also used to get mixed up with Pethia kuminji, who has two spots, but they actually look like vertical lines. And also sometimes, now when we were going through the survey, I was going through the area to see whether there were other locations where the Bandula barb could be found. And sometimes I was given messages by the villagers to come and see that there are similar fish with 2 lines, to see that they were the fingerlings of Pantius singhalea. And in these 2 also, the extra scale is there for the nigrofasciatus, but there is no extra scale for Pethia kuminji. Next slide, please. The location, the fish is found in a small stream at Galapitamada in the Kegol district. And the stream originates from Rabbedigala. And it flows through the village of Rabbedigala, Hapugoda, and Minipura villages and falls into the Gurugoda Oya, which then goes into the Kalani River basin. In Pethiagoda's paper in 1991, he had described saying that the fish was restricted to 500 meters of the stream, which is probably the Minipura area where his work was conducted. But during my study, I discovered that it actually stretched to 2.5 kilometers, that is from Rabbedigala to Minipura. Then you would wonder, why does it not go beyond that? That is simply because at the end of the stream, there are huge boulders, and then it goes into a rubber plantation, and the substratum and the fish assemblage, they are all different. So that is probably why the fish is not distributed beyond that point. And the most important aspect of this location is that it is outside the protected area network. So that is why you need the community to help us to conserve this fish species. Next slide, please. This is a type locality of P.bandula, and the stream that it likes is actually shallow, and it has the Ketala plants and Kumbuk growing on the sides. And one side of the stream contains home gardens, and the other side contains paddy fields. And the water is not deep at all. And also the flow rate is very slow. Next slide, please. This figure also shows a part of the stream and where the Bandula barb, where it's its habitat. So here you can clearly see the paddy field on one side, stream on one side, and on either sides you get rubber plantations, coconut plantations, and home gardens. Next slide, please. These are also some places in the type locality where the Bandula barb was found. And an interesting thing to note is that if you see the picture below, you can see part of the stream on the right-hand side and a big pool on the left. The story behind it is that this is a breeding pool. So during the dry season, there's a small channel which leads from the stream to the pool. And there we found that the Pantius bandula, the female, goes there and lays his eggs, and fingerlings are born there. And during the same time, I saw little shrimps. So I assumed that probably the fingerlings fed on the shrimps during that time. And when it starts to rain, the pool fills up, and then it links with the main stream, and then the fingerlings and the adults go back to the stream. So that's a very interesting thing that we observed. And also now the site on the left, even though it looks it looks as if it's a very large stream, when the dry season comes, most of the parts of the stream actually dry up. They actually shrink to quite a narrow strip, and also the water becomes extremely shallow. Next slide, please. The identified threats, the anthropogenic threats that we saw, was that the stream tends to become narrow when people start their paddy cultivation. In the first photograph, you can see that the stream is on the far left corner, and it is quite shrinking also. And in the other photographs, you can see how home gardens, how parts of the stream gets encroached by the home gardens. And also the rubber plantations, they come right to the edge of the stream, and then they are cleared. The Ketala, the Kumbuk, all those plants that support the habitats of the Pethia bandula are then removed. So then it has no place to hide. And sometimes when the flow rate becomes high, the flow rate around the fibrous roots of the Ketala and the Kumbuk are very low. So then they can hide there. And also the little fingerlings also hide in the fibrous roots of the Ketala and the Kumbuk. So next slide, please. There were also instances where parts of the stream were cemented. So no matter what the villagers said, sometimes the irrigation people come and then they had cemented part of the stream. Luckily though, there had been a very big shower of rain, and part of the stream, part of the cemented area had collapsed. So it has now come back to normal. And there are also instances where agrochemicals, fertilizers are also dropped into the stream. And we were also very worried because the key area where the watershed starts, that is also an area where you get home gardens. So sometimes it's a possibility of encroachment. Further, there were instances where the kitchen waste was led from a pipe and led straight into the stream. So where the water got polluted, and especially during the dry season, this affects the fish quite badly. Next slide, please. So as I mentioned before, the narrowing of the stream, and also there were instances where there was an illegal collection of fish by unauthorized parties. And also some of the people, they link the pipes from the stream to their homes because they do not have a pipe water system. And most of the time, the water goes waste because of the pressure, they keep the taps open. And then what happens is large quantity of water goes waste. And then during the dry season, the stream dries up quite quickly. So those are also some of the anthropogenic threats that we observed in this area. Next slide, please. And considering the population size dramatically declined during the past 2-3 decades. So much so that when I was doing the survey, I could count only about 123 fish. But of course, there could have been plus or minus, but it had really drastically declined during the years. Next slide, please. During my MSc program, another area that I studied was the previous translocation done in 2001. So this was done after the fish was declared as a protected species, and there was a fish exporter called Mr. Ananda Patirana, and he had captively bred the P.bandula barb. So the YZA and the Department of Wildlife got together with Mr. Patirana, and they decided to introduce it to a stream at Salgala. So these are the pictures of Salgala. And you can see that actually the substratum is extremely rocky. And during the dry season, it dries up. So only there was a pool on the right-hand side. That was actually one place where the barbs were observed. So they introduced 250 captively bred pairs into the stream in 2001. But when I went in 2003, 2004, there were very few fish species over there. And also the fish assemblage was different because here I found the Bulla thapea and the Belontia signata. Next slide, please. So the individuals with mixed traits were the barbs that I saw in this pool at Salgala. So from these pictures, you can see they look very mixed because they don't have the typical characteristics of the P.bandula barb, but they have mixed characteristics like blotches, extra blotches, and complete lateral lines, and some don't have extra scalation. So those were quite different to the original P.bandula barbs. So what I did was I also visited Mr. Patirana, and he had been keeping one set of barbs as a sample. And from that, I could see that those barbs were also like this. So probably down the line, they would have got mixed. So one good thing that happened in this program was that they had introduced it to a separate stream, which did not link to the stream at Galapitamada. So that way, the type locality was not contaminated. Next slide, please. So I'm just putting up this graph which shows the site disparity, because when you actually do translocations and the features of the stream have to match the type locality. So during my study, I was able to collect a lot of information on the physical, chemical characteristics of both streams. And when this study was done, it quite clearly showed that Salgala differed greatly with Galapitamada. And also the features that they differed in greatly was the substratum, the fish assemblage, and the depth of the water column. So after this program, after my study, when we realized that the number of P.bandula barbs had declined greatly, we spoke with the Ministry of Environment, and with Professor Devaka leading the program, we designed a recovery plan for the P.bandula barb. And there, the key components was to actually look for a similar location where it had the similar characteristics, the physical, chemical characteristics, similar fish assemblage as that of Galapitamada, so that we could introduce a population there, and also so that we could begin, and we actually found a place at Wilkulakanda, which was quite similar to the stream at Galapitamada. And also the other purpose of the recovery plan was also to design a community development program. So especially since this is not a site which is in a protected area. So in order to protect this fish species, we must involve the community. Without the help of the community, it is very difficult to conserve a fish species like this. Next slide, please. Yeah. So the rest of the program will be shown by Mr. Tharaka Muthunayake, my colleague, and he will describe the community-based conservation program. Thank you, and over to you, Tharaka.
Thank you, Hasula. Now we are moving to community-based conservation program. So I think all of you got understood that the threat faced by the fish throughout a few decades before starting the project. So therefore, we, as described by Hasula, we started to implement community-based conservation program. So here, you can observe, compared to 1980s and 1990s, there was a huge declining of the fish population. But I think you observed that in 2003, there were only about 100 or just over 100 fish individuals. But when you started the project in 2010, we recorded 260 individuals. At the beginning of the conservation program, population size has increased up to 262. But if we consider with the previous or past, there was a huge declining of the population size. It is also one of the reasons to implement community-based conservation program. Actually, we wanted to go to an in-situ conservation program at the type locality. So here, this is the overall objective of the program. So actually, we want to ensure the long-term survival of P.bandula in its type locality through participatory conservation approach and establishment of a second population in a new habitat. Those are the overall objectives. So we identified specific objectives under this overall objective. So here you can observe those specific objectives in the slide. First specific objective was to assess the degree of awareness and willingness of the community to conserve P.bandula or Pethia-bandula. So it was a must, because when we move to community-based conservation program, actually we want to know the willingness of the community towards this conservation effort. Without involvement of the community, it was a failure. Therefore, first we wanted to identify the willingness and the degree of awareness of the community. Then the second objective was to create awareness within the community about the need of conserving P.bandula. Then third was to establish a community-based conservation program for Pethia-bandula, so for a long-term survival of the species. Finally, the final objective was translocation of Pethia-bandula to a new locality to establish a second population. So we will move one by one. Then I will simply explain how did we manage to achieve these objectives. Okay, first we are moving to the first objective, that is about determining the degree of awareness and willingness of the community to conserve Pethia-bandula. So we actually identified or determined the degree of awareness using a questionnaire survey. So we carried out 42 randomly selected families. That means questionnaire survey was carried out in 42 randomly selected families surrounding the stream at Hapugoda and Alpitiya. So this was carried out to determine the degree of awareness, to determine the willingness of the community to conserve Pethia-bandula, and to obtain a profile of the community. So we actually wanted to know their living status, then their economic status, and also their social activities. So it was very important. Those were very important in conserving species in their type locality. And also we use this questionnaire to introduce the objectives of the program. Clear? Then here we determine the degree of awareness of the community like this. Awareness was determined based on 8 questions, and a score was given for correct answers as a percentage for each questionnaire. So finally, people who scored equal or above 75% were considered as those who were aware. Then this is the result of the questionnaire. Here we found that the degree of awareness of the community at the beginning of the program was 54%. So as analyzed by questionnaire, we found that 38 out of 42, that means around 90.4% of the community was keen to protect the species via community program. Then around 85%, that means 36 out of 42 of them agreed to actively participate in the conservation program. So actually we address these things in the questionnaire survey. Therefore, we concluded with these percentages. Then after determining the willingness of their community to actively participate in the conservation program, so we moved to our second objective. That was creating awareness within the community about the need of conserving Pethia-bandula. So under this, several community awareness workshops were carried out. So actually 2 community awareness workshops were carried out at Hapugoda and Alpitiya with the participation of Divisional Secretariat of Tharaka. So here you can see these are some of the pictures which were taken during the community workshop at Hapugoda. Actually, people were actively participated in these community programs. So we Divisional Secretariat and Professor Devaka and also different other resource persons gave lectures and awareness about the awareness of P.bandula, Pethia-bandula here. These are some of the pictures taken during the community workshop of Alpitiya. Okay, then as a second tool of creating awareness or increasing the awareness among the community, we use a brochure. So that means other than the awareness programs, we use different communication tools that were used to increase the awareness. One is this brochure. So this brochure includes a brief description of the species, its habitat, decline in the population over past years, and the threats that have contributed to its decline, importance of their village, and the ways to protect Pethia-bandula and its habitat. So these were the major components of the brochure. So we distributed this brochure among every villager, or that means we visited their houses and distribute this. And also these were distributed among children in the community and also among schools to increase the awareness. Then also we prepared 2 calendars for 2011 and 2012. We started the program in 2010. Then 2 calendars were prepared for 2011 and 2012. So 2011 calendar, here you can observe this is the 2011 calendar. So in this describe the P.bandula, Pethia-bandula and its habitat and the conservation program. So through this also we wanted to convey the message of to community. Actually, this was a hit. So after about 5 or 6 years, we visited the same houses. Then we observed they kept this calendar, also the 2012 calendar. They kept these 2 calendars at their home. They still love this calendar. So therefore, we can say this was a hit. We were able to convey the message to the community mainly through this calendar because they love this. Then here you can observe this is the 2012 calendar. This describes the overall biodiversity and the historical importance of the area and the measures to protect biodiversity, emphasizing Pethia-bandula. Then other than above, presentations and video clips prepared on Pethia-bandula and its habitat were used as communication tools to enhance awareness. So here are some snapshots or images taken from those video clips. So these were played in community workshops, during community workshops, and also during the community meetings, during the discussions with them, we played these video clips and made some discussions. In addition, we made a transect walk. Transect walk was done with the community members along the stream to identify threats to the species by themselves. Then we are moving to the third objective. After the awareness program, we move to our third objective. it was establishing a community-based conservation program for P.bandula. This is one of the key events in our community program. So under this, as a first event, we established an environment protection committee. So this was established with the cooperation of the community members of Hapugoda, Galapitamada, and Alpitiya villages. Awareness was further raised among the committee through presentations and discussions. Then action plan to protect the species was drawn up. Yes, we made some discussions with the community members and made an action plan using their ideas as well. Then second was we set 2 signboards in the village. We set 2 signboards. So you can see this slide represent the content of the signboard. It also includes basic information about P.bandula and its awareness. Here, this provides a message. Actually, this provides a message about conservation of P.bandula and its habitat. This was set near the stream, the entry point to the Hapugoda village. Such signboards were set. Here, then, when setting the signboards, also we actually got the participation of the community. So actually, they love this effort and they actively participated in setting the signboard. So here you can observe these are some of the pictures taken during setting the signboard in their village. Okay, then additionally, monthly community meetings with the environment committee was conducted. So as I told you earlier, during these community meetings, we made some discussions. They were nice discussions, and also we play some video clips about biodiversity and also about the P.bandula. So likewise, community meetings were conducted. Then also timetable on freshwater fish of Sri Lanka was prepared. So then actually this was prepared to distribute among school children. Here you can see later, we did some school environmental programs as well. So we distribute this timetable among villages where school children are there, and also we distributed this timetable among school children. So here it provides a concise description on freshwater fishes in Sri Lanka, their distribution and the conservation measures. Okay, because here the purpose of preparing this timetable was increasing the awareness among school children, because for the sustainability of this project, we identified that children play an important role. Therefore, we wanted to first most of the elders or the most of the elders in the society involved in community programs or involved in community meetings. But later we identified that and we wanted to gather young children and young ones. So therefore, to gather to attract young children, school children to our program, we did some activities like this. Okay. Then additionally, we did a tree planting in the habitat, and also we distribute plants among villages mainly. So this was a habitat enrichment. There were some places. Hasula said that mainly Kumbuk and Ketala were prominently found in their habitat. So between fibrous roots of Ketala, the fingerlings and the youngs of P.bandula hide during the rainy season as well. Okay, therefore, so we wanted to then also be noticed that there were some places in which stream was collapsed. We selected such areas and planted some trees, as well as we distribute someone asking to set my camera. So here I don't know why it is not working. I have switched on my camera on color TNA, but
we can see you, Tharaka. No issue. You can continue.
Okay, then it's okay. Fine. Okay. And
you can see the top of your head. Okay, now we can see you.
Okay. Okay. Then here we did a tree planting in the habitat, and we distributed plants among the villages, mainly Kumbuk and Ketala, that are important to provide microhabitat for P.bandula were planted along the stream with the participation of the community members. Here you can see these are some of the community members who are actively involved in tree planting program. Then, as I told you, we distributed some plants. We distributed some plants among villages, mainly some fruit plants and some butterfly host plants were also distributed among villages. So actually, we asked the plants that required by these villages. So we made a list. Actually, we made some discussions with villages and asked whether they need these plants and which type of plants are required. Because there were elders who aware about the history of the village. They said that these plants were there in history, but now they are not observed. So likewise, we made some discussions and distributed those required plants among villages, along with the fruit plants and some butterfly host plants. Okay, then next activity was, as I told you, school environmental programs. So we conducted 3 school environmental programs at 3 adjacent schools of the area. So as I told you, the main purpose of this was get the attraction of the school children towards this conservation effort. So these are some topics that we discuss in the environmental programs: the biological diversity of Sri Lanka, the biological diversity of their villages, then the environment conservation, ecosystems, then and also P.bandula and its conservation. So there were some resource persons, Aruna Padma Peruma, Sampath Gunatileka. Those were main resource persons conducting these environmental programs. So as I told you, we conducted environmental programs in 3 schools. Those are Balagamba Mahavidyalaya Galapitamada, Kadigamu Primary School Galapitamada, and also Kiuldeniya Mahanaga Mahavidyalaya Galapitamada. These are some adjacent or surrounding schools in the area. Most of the children in Hapugoda and Rabidigala area or Alpitiya area study in these schools. Okay, here you can see these are some pictures taken during the school environmental programs. Okay, then at the end of the conservation program, at the end of the awareness program, actually at the end of the awareness program and the community-based conservation, we actually judge the awareness here using structured interviews among villages. As judged by using structured interviews, we judge the awareness. We determine the awareness. As judged by structured interviews, we observe that the degree of awareness had increased up to 85% end of the awareness program. So and at the end of the awareness campaign, it was observed that the attention of the community on the stream and P.bandula was increased. Then that means, so it was reflected by minimization of the anthropogenic threats. That means when we made discussions earlier, they didn't bother about the stream. They bother only about their cultivations and their home garden. But after the awareness program, they always talked about the fish. They always talked about the conservation of the stream. They minimize the anthropogenic threats. So we clearly observed that. Then population size of P.bandula gradually increased, most probably with the reduction of the anthropogenic threats. Here you can observe, so this graph represents the variation of population size of P.bandula over time at locality. Here you can observe, we started the project in 2010. Then during the conservation program, population is almost stable. So before that, we can observe there were fluctuations. Like here, this is the point, or in February 2010, this was our initial count at the beginning of the conservation program. We said that I told you that there were 262 individuals recorded. Then you can observe during 2010 there were fluctuations, but in 2011 mainly we observe static or stable population. This is the period where our conservation program was implemented. And after the conservation program, you can clearly observe here there was an increase in the population size. So we thought that this mostly should be due to reduction of the human threats or anthropogenic threats. Okay, then we are moving to the last objective. It was translocation of a small stock of P.bandula to establish a second population. The reason to had such a decision was because the type locality of Pethia-bandula was outside the protected area network. So therefore, we wanted to translocate it. We want to translocate small stock of Pethia-bandula into a forest reserve or into a protected area network. So also we have seen that Hasula said that the previous translocation carried out by YZA with the cooperation of Department of Wildlife Conservation was actually a failure. So therefore, we wanted to carry out proper translocation. So then here simply I will explain how we did this translocation program. Here, if I mention about a new locality, it's like this. This is a stream that flows through Wilkulakanda Forest Reserve, found in Botale Ihelagama, Warakapola district. So this is the location. Then actually this stream flows to Maoya. This stream flows to Maoya. That's its locality. Then we monitor the before introducing small stock of Pethia-bandula to the new locality. Actually, we did a monitoring program. We monitor the new locality throughout 1 year during the rainy season as well as during the dry season. We monitor the new locality. Then these water quality parameters were monitored, such as flow rate, water depth, pH, salinity, nitrate levels, phosphate levels, dissolved oxygen levels, turbidity. We identified these as important water quality parameters for the survival of the fish. In addition, we monitored fish composition, floral assemblages of the original and selected site for the translocation. We actually matched the fish composition and the floral assemblages over a period of 1 year in 2011. Actually, this was implemented simultaneously with the conservation program. In addition to that, presence of microhabitats, availability of food, and the possible threats were monitored at the new site. And further discussions were held with the two communities, Hapugoda and Ihelagama. Ihelagama means the new locality, about the translocation program. So that means simply this was also carried out with the participation of the community. We got the participation of members of both villages. Okay. Here, this table summarizes some water quality parameters in the type locality and the translocated site. The type locality is a Galapitamada, and this is the translocated site, Wilkulakanda. You can see here, most of these water quality parameters are same. Then here you can see there's a significant difference in turbidity, dissolved oxygen concentration, water temperature, flow rate, almost same in both sites. But there's a high turbid condition in the, if you consider turbidity of water, in the stream of type locality of Galapitamada, there was a high turbidity. But Wilkulakanda stream was much clear. And also you can observe nitrate and phosphate concentration in Galapitamada stream or type locality stream was comparatively significantly high. The most possible reason for this high nitrate and phosphate content may be usage of fertilizers. And also then we said that, I think you observed that the stream flow through agricultural areas, mainly through paddy fields. Therefore, then it may be reason for the high turbidity. But Wilkulakanda stream was completely found inside the protected area network. So therefore, there were low nitrate and low phosphate concentrations. And these are the fish assemblages. You can see almost similar species found in both Galapitamada and Wilkulakanda. Clear? So here there was some different. The difference is regarding Belontia signata. Belontia signata was recorded in Wilkulakanda, that means our new locality. But in 2011, we didn't record or we didn't find Belontia signata in Galapitamada. Belontia signata in Thalkosa was not found in Galapitamada. So, but we heard that even in Galapitamada there were Belontia signata. Clear? We heard that there were Belontia signata. First, we thought that Belontia signata, Belontia signata is somewhat aggressive fish species, not that much, but it's somewhat aggressive. Therefore, we thought that it may be a threat to Pethia-bandula, but actually it was not. So we later observed that actually we didn't select the sites where Bellontia signata were present to translocate or to introduce Pethia-bandula. But later, after many years, we observed that Pethia-bandula actually coexist with Bellontia signata. There was no threat from Bellontia signata. There is no attack because Pethia-bandula is not a territorial fish. So therefore, they coexist. So it was a success. Then after monitoring these parameters, we observed that new habitat or new locality was suitable for translocation. Then we introduced 5 adult males and 15 adult females to a new site on February 2012 with the participation of the community. Here you can observe community members from both villages release this small stock of fish to new locality. And also a volunteer to monitor the new habitat was appointed from the community of the new site. Then we do backside counts. Backside counts were taken at 3 months, 6 months, and 1 year, and 3 years after translocation. Then here we recorded after translocation, we recorded 10 adults and 7 fry were observed in the new locality 3 months after translocation. That means here we introduced 20 adults collectively. Total number of fish introduced were 20. But after 3 months, we observed 10 adults. And the most important thing was there were 7 fries. That means it indicates that the fish, introduced fish, has reproduced. So they had bred in the new site. So therefore new fries were not introduced, but we observed fries. That means at least 1 breeding cycle should have happened within these 3 months. So it indicates the suitability of the site. Clear? That's why we observe breeding. Also, we noted that after 3 months, they have distributed 15 meter upstream and 20 meter downstream from the point of translocation after 3 months. You can observe this is adult female fish at the translocated site, and this is an adult male fish at the translocated site. Then these are the counts taken 6 months after translocation. So total of 26 individuals, 5 fries, fingerlings, 5 adult males and 6 adult females were recorded after 6 months. Among them, 3 fingerlings were distributed 20 meter upstream, while 7 adults and 3 fry were distributed 20 meter downstream from the point of translocation. Therefore, you can observe this is the total number of fish that we have translocated, 15 females and 5 males. Then after 6 months, there was an increase in population size. Mainly, we recorded, more interestingly, we recorded fingerlings and fry. That means they had bred in that site. So compared to previous translocation done by DWC and YZA, actually this was a success because we noticed that here this table summarizes the failure of the previous translocation. I think Hasula said that how much the Salgala site was different compared to type localities. The main reason for this declining of the population. Here, Salgala site, after 3 months translocation, only 30 fish were observed. Then after 6 months, 20 fish. But initially 500 individuals were introduced to that site. Then after 13 months, 15, and after 3 years, they had declined up to 10 fish. But such a record was not observed in new translocation. So here you can see this is the variation of population size of Pethia-bandula translocated site. So this is the in February 2012 is the was the time where when we did the translocation. This is after 3 months, then this is 6 months. Then after 1 year, you can observe total population size was 68. So population size has increased up to 68. The translocated population had distributed along 1 kilometer of the stream after a year. So that means from the site of translocation, they have distributed over 1 kilometer. Then after 3 years, we observed that we counted 270 individuals in the new locality or translocated site. So you can see here, therefore, population size has increased over 3 years. Okay, then here actually we made a final visit, last visit during a rainy season, during a rainy day actually. So after this visit, we couldn't visit the site due to this fuel shortage and transportation problems, you know. But during our last visit that we did several months earlier, actually, unluckily, it was a rainy day. Therefore, the entire water stream was turbid. So you can see the water was turbid and had a high flow rate like this. This is a translocated site. Therefore, making counts was successful. However, even though water is this much turbid, we recorded 35 individuals. Also, we recorded that fish had distributed over 2 kilometer stretch downstream from the site of translocation. Here, fry and fingerlings were recorded at 2 kilometers downstream, which indicates breeding. According to observations, it had established well in the stream. So, even with this turbidity, we recorded 35 individuals. Definitely, there should be more. Clear? Okay. Then here, this is the acknowledgement. I can acknowledge like this. Then, thank you for listening up.
Thank you very much, Hasula and Tharaka, for that very interesting talk. We have over the years heard so much about translocation of elephants. I think this is the first time that we have had this a talk about translocation of another species, about fish. And it's good to recognize that the importance of the environment in the locality to which the animals are going to be transferred, translocated, as well as their natural habitat. And also very important, the community involvement. And I think it was quite, it would have been very interesting for most of our members to hear how you set about it and had the discussions with the community and so on. So the one question that remains in my mind is about the sustainability of this. I know that visiting the site in the recent past has been difficult, but do you have an ongoing program to monitor the sites, both the Galapitamada site as well as the new site, to see that your good work has not been wasted, but it's still
Yes.
somebody
Dr. Malik,
Yeah.
yes, Dr. Malik, now our program is we actually, all this while after the funding from the Biodiversity Secretariat, we